Neuron is similar to any other cell in the body, having nucleus and all the organelles in cytoplasm. However, it is different from other cells by two ways. Neuron has branches or processes called axon and dendrites. Neuron does not have centrosome. So, it cannot undergo division. Classification of neuron. Neurons are classified by three different methods. Depending upon the number of poles, depending upon the function, depending upon the length of axon. Depending upon the number of poles, based on the number of poles from which the nerve fibers arise, neurons are divided into three types: unipolar neurons, bipolar neurons, multipolar neurons. Unipolar neurons are the neurons that have only one pole. From a single pole, both axon and dendrite arise, Fig 134.1. This type of nerve cells is present only in embryonic stage in human beings. Bipolar neurons with two poles are known as bipolar neurons. Axon arises from one pole and dendrites arise from the other pole. Multipolar neurons are the neurons which have many poles. One of the poles gives rise to axon and all other poles give rise to dendrites. Depending upon the function, on the basis of function, nerve cells are classified into two types: motor or efferent neurons, sensory or afferent neurons. Motor or efferent neurons are the neurons which carry the motor impulses from central nervous system to peripheral effector organs like muscles, glands, blood vessels, etc. Generally, each motor neuron has a long axon and short dendrites. Sensory or afferent neurons are the neurons which carry the sensory impulses from periphery to central nervous system. Generally, each sensory neuron has a short axon and long dendrites. Depending upon the length of axon, neurons are divided into two types: Golgi type I neurons, Golgi type II neurons. Golgi type I neurons have long axons. Cell body of these neurons is in different parts of central nervous system and their axons reach the remote peripheral organs. Golgi type II neurons of this type have short axons. These neurons are present in cerebral cortex and spinal cord. Structure of neuron is made up of three parts: nerve cell body, dendrite, axon. Dendrite and axon form the processes of neuron, Fig 134.2. Dendrites are short processes and the axons are long processes. Dendrites and axons are usually called nerve fibers. Nerve cell body is also known as soma or perikaryon. It is irregular in shape. Like any other cell, it is constituted by a mass of cytoplasm called neuroplasm, which is covered by a cell membrane. The cytoplasm contains a large nucleus, Nislo bodies, neurofibrils, mitochondria, and Golgi apparatus. Nislo bodies and neurofibrils are found only in nerve cell and not in other cells. Nucleus: each neuron has one nucleus, which is centrally placed in the nerve cell body. Nucleus has one or two prominent nucleoli. Nucleus does not contain centrosome. So, the nerve cell cannot multiply like other cells. Nislo bodies or Nislo granules are small basophilic granules found in cytoplasm of neurons and are named after the discoverer. These bodies are present in soma and dendrite but not in axon and axon hillock. Nislo bodies are called tigroid substances, since these bodies are responsible for tigroid or spotted appearance of soma after suitable staining. Dendrites are distinguished from axons by the presence of Nislo granules under microscope. Nislo bodies are membranous organelles containing ribosomes. So, these bodies are concerned with synthesis of proteins in the neurons. Proteins formed in soma are transported to the axon by axonal flow. Number of Nislo bodies varies with the condition of the nerve. During fatigue or injury of the neuron, these bodies fragment and disappear by a process called chromatolysis. Granules reappear after recovery from fatigue or after regeneration of nerve fibers. Neurofibrils are thread-like structures present in the form of network in the soma and the nerve processes. Presence of neurofibrils is another characteristic feature of the neurons. The neurofibrils consist of microfilaments and microtubules. Mitochondria are present in soma and in axon. As in other cells, here also mitochondria form the powerhouse of the nerve cell, where ATP is produced. Chapter 1. Golgi apparatus of nerve cell body is similar to that of other cells. It is concerned with processing and packing of proteins into granules. Chapter 1. Dendrite is the branched process of neuron, and it is branched repeatedly. Dendrite may be present or absent. If present, it may be one or many in number. Dendrite has slow granules and neurofibrils. Dendrite transmits impulses towards the nerve cell body. Usually, the dendrite is shorter than axon. Axon is the longer process of nerve cell. Each neuron has only one axon. Axon arises from axon hillock of the nerve cell body, and it is devoid of slow granules. Axon extends for a long distance away from the nerve cell body. Length of longest axon is about 1 meter. Axon transmits impulses away from the nerve cell body. Organization of nerve. Each nerve is formed by many bundles or groups of nerve fibers. Each bundle of nerve fibers is called a fasciculus. Coverings of nerve. The whole nerve is covered by tubular sheath, which is formed by a areolar membrane. This sheath is called epineurium. Each fasciculus is covered by perineurium, and each nerve fiber, axon, is covered by endoneurium. Fig 134.3. Internal structure of axon. Axocylinder. Axon has a long central core of cytoplasm called axoplasm. Axoplasm is covered by the tubular sheath-like membrane called axolemma. Axolemma is the continuation of the cell membrane of nerve cell body. Axoplasm along with axolemma is called the axocylinder of the nerve fiber. Fig 134.4. Axoplasm contains mitochondria, neurofibrils and axoplasmic vesicles. Because of the absence of slow bodies in the axon, proteins necessary for the nerve fibers are synthesized in the soma and not in axoplasm. After synthesis, the protein molecules are transported from soma to axon, by means of axonal flow. Some neurotransmitter substances are also transported by axonal flow from soma to axon. Axocylinder of the nerve fiber is covered by a membrane called neurolemma, see below. Non-myelinated nerve fiber described above is the non-myelinated nerve fiber, which is not covered by myelin sheath. Myelinated nerve fiber which is insulated by myelin sheath is called myelinated nerve fibers. Myelin sheath is a thick lipoprotein sheath that insulates the myelinated nerve fiber. Myelin sheath is not a continuous sheath. It is absent at regular intervals. The area where myelin sheath is absent is called node of Ranvier. Segment of the nerve fiber between two nodes is called internode. Myelin sheath is responsible for white color of nerve fibers. Chemistry of myelin sheath. Myelin sheath is formed by concentric layers of proteins, alternating with lipids. The lipids are cholesterol, lecithin, and cerebroside, sphingomyelin. Formation of myelin sheath. Myelinogenesis. Formation of myelin sheath around the axon is called the myelinogenesis. It is formed by Schwann cells in neurolemma. In the peripheral nerve, the myelinogenesis starts at 4th month of intrauterine life. It is completed only in the 2nd year after birth. Before myelinogenesis, Schwann cells of the neurolemma are very close to axolemma, as in the case of unmyelinated nerve fiber. The membrane of the Schwann cell is double-layered. Schwann cells wrap up and rotate around the axocylinder in many concentric layers. The concentric layers fuse to produce myelin sheath, but cytoplasm of the cells is not deposited. Outermost membrane of Schwann cell remains as neurolemma. Nucleus of these cells remains in between myelin sheath and neurolemma. Functions of myelin sheath. Faster conduction. Myelin sheath is responsible for faster conduction of impulse through the nerve fibers. In myelinated nerve fibers, the impulses jump from one node to another node. This type of transmission of impulses is called saltatory conduction, chapter 136. Insulating capacity. Myelin sheath has a high insulating capacity. Because of this quality, myelin sheath restricts the nerve impulse within single nerve fiber and prevents the stimulation of neighboring nerve fibers. Neurolemma. Neurolemma is a thin membrane, which surrounds the axis cylinder. It is also called neurolemmal sheath or sheath of Schwann. It contains Schwann cells, which have flattened and elongated nuclei. Cytoplasm is thin and modified to form the thin sheath of neurolemma. One nucleus is present in each internode of the axon. Nucleus is situated between myelin sheath and neurolemma. In non-myelinated nerve fiber, the neurolemma surrounds axolemma continuously. In myelinated nerve fiber, it covers the myelin sheath. At the node of Ranvier, where myelin sheath is absent, neurolemma invaginates and runs up to axolemma in the form of a finger-like process. Functions of neurolemma in non-myelinated nerve fiber. The neurolemma serves as a covering membrane. In myelinated nerve fiber, it is necessary for the formation of myelin sheath, myelinogenesis. Neurolemma is absent in central nervous system. So, the neuroglial cells called oligodendroglia are responsible for myelinogenesis in central nervous system. Neurotrophins, neurotrophic factors. Neurotrophins or neurotrophic factors are the protein substances, which play an important role in growth and functioning of nervous tissue. Source of secretion. Neurotrophins are secreted by many tissues in the body, particularly muscles, neuroglial cells called astrocytes and neurons. Functions. Neurotrophins facilitate initial growth and development of nerve cells in central and peripheral nervous system, promote survival and repair of the nerve cells, play an important role in the maintenance of nervous tissue and neural transmission. Recently, it is found that neurotrophins are capable of making the damaged neurons regrow their processes in vitro and in animal models. This indicates the possibilities of reversing the devastating symptoms of nervous disorders like Parkinson's disease and Alzheimer's disease. Commercial preparations of neurotrophins are used for the treatment of some neural diseases. Mode of action. Neurotrophins act via neurotrophin receptors, which are situated at the nerve terminals and nerve cell body. Neurotrophins bind with receptors and initiate the phosphorylation of tyrosine kinase. Types. Nerve growth factor, NGF, was the first protein substance identified as neurotrophin. Now, many types of neurotrophic factors are identified. Nerve growth factor, NGF, is a neurotrophin found in many peripheral tissues. Chemistry. NGF is a peptide with 118 amino acids. Each molecule of NGF is made up of 2 alpha subunits, 2 beta subunits, and 2 gamma subunits. Only the beta subunits have nerve growth stimulating activity. Functions. NGF promotes early growth and development of neurons. Its major action is on sympathetic and sensory neurons, particularly the neurons concerned with pain. Because of its major action on sympathetic neurons, it is also called sympathetic NGF. NGF also promotes the growth of cholinergic neurons in cerebral hemispheres. Commercial preparation of NGF extracted from snake venom and submaxillary glands of male mouse is used to treat sympathetic neuron diseases. NGF plays an important role in treating many nervous disorders such as Alzheimer's disease, neuron degeneration in aging, and neuron regeneration in spinal cord injury. Other neurotrophins. Brain-derived neurotrophic growth factor. Brain-derived neurotrophic growth factor, BDGF, was first discovered in the brain of pig. Now it is found in human brain and human sperm. BDGF promotes the survival of sensory and motor neurons, arising from embryonic neural crest. It also protects the sensory neurons in peripheral nervous system and motor neurons of pyramidal system. It enhances the growth of cholinergic, dopaminergic, and optic nerves. It is suggested that BDGF may regulate synaptic transmission. Commercial preparation is used to treat motor neuron diseases. Ciliary neurotrophic factor, CNTF. CNTF is secreted in peripheral nerves, ocular muscles, and cardiac muscle. It protects neurons of ciliary ganglion and motor neurons. Glial cell line-derived neurotrophic factor, GNDF, GDNF is found in neuroglial cells. It has a potent protective action on dopaminergic neurons. It is used for the treatment of Parkinson disease. Fibroblast growth factor, FGF. FGF was first discovered as growth factor promoting the fibroblastic growth. It is also known to protect the neurons. Neurotrophin 3, NT3, neurotrophin 3, NT3, acts on gamma motor neurons, sympathetic neurons and neurons from sensory organs. It also regulates the release of neurotransmitter from neuromuscular junction. NT3 is useful for the treatment of motor axonal neuropathy and diabetic neuropathy. Recently, few more substances belonging to the neurotrophin family such as NT4, NT5, and leukemia-inhibiting factor are identified. NT4 and NT5 act on sympathetic neurons, sensory neurons, and motor neurons.